Haemopoiesis. Haemopoiesis, also known as haemopoiesis, is the formation of the cellular components of blood. Hemato or hemo means blood and poiesis means formation or production. The hematopoiesis can be classified into two broad categories depending on the site in which it occurs. It can be either medullary or extra, that is outside medulla, extramedullary. Medulla is basically referring to the bone marrow, especially the red bone marrow. So, medullary hematopoiesis occurs in the red bone marrow of the bones of the axial skeleton, such as the proximal ends of the epiphysis of long bones such as femur and humerus, the pelvic and shoulder girdles, and the sternum. Whereas the extramedullary, that is occurring outside the bone marrow, occurs in organs such as the liver, the spleen, lymph nodes and thymus. Now, some fun facts about the medullary hematopoiesis. Medullary means occurring in the medulla or red bone marrow. What is the red bone marrow? Red bone marrow is the connective tissue present in the microscopic spaces between the trabeculae of bone tissues. So, bone tissues are divided by spaces and within these spaces is the marrow. This is the red bone marrow. Now, as adult bodies mature, the rate of formation of blood decreases. So, the red blood marrow, which is active, gets converted into a more stable inactive color and becomes yellow bone marrow, which is filled with more fat cells. However, when conditions such as purpura or bleeding occur, what happens is the yellow bone marrow becomes active once again and it gets converted back into red bone marrow. Sites of hematopoiesis. As we have discussed, medullary hematopoiesis is predominant. However, depending on the different junctures of life cycle, the site of hematopoiesis differs. In an embryo, that is up to about 3 months of age, the primary site of hematopoiesis is the yolk sac. Then, in the fetus, the primary site of hematopoiesis are the liver and spleen. However, after the about 6 months, that is the third trimester of pregnancy, what happens is the red bone marrow becomes the primary site of hematopoiesis and this continues until adulthood. So, in an adult body, the red bone marrow of the bones of the axial skeleton, which we have discussed, is the primary site of haemopoiesis. However, sometimes the liver and spleen can also contribute to haemopoiesis even in the adult body. This happens during any kind of pathological states or infections. The process of hematopoiesis always begins with a pluripotent stem cell. Pluripotent means capable of many functions. Now, this pluripotent stem cell can give rise to lymphoid progenitors or rather and myeloid progenitor line. Now, the lymphoid progenitor line and the myeloid progenitor lines are together known as multipotent or colony forming units because these form various clusters of cells. Hence, the progenitor cells are also known as colony forming units. What happens next is the progenitor cells then divide into precursors of the mature blood cells. Now these precursors are called blasts. So the lymphoid progenitor will divide into 3 types of lymphoblasts. On the other side, the myeloid progenitors are dividing into the erythroblast, the megakaryoblast, and band neutrophils, band eosinophils, and band basophils. It also divides into promonocyte as well as mast cell precursor. In the next stage, these blasts, which are all precursors to the final blood cells, divide into, this lymphoblast divides into the pre-T cell, naive B cell, natural killer cell. Another cell called the dendritic cell also comes from the lymphoid progenitor. On the other side, megakaryoblast gives rise to platelets, promonocyte gives rise to monocyte, mast cell precursor gives rise to mast cells, and band neutrophils, band eosinophils, and band basophils give rise to neutrophils, eosinophils, and basophils respectively, and the erythroblast gives rise to the RBC. So as you can see, the first step in the formation of the blood is the pluripotent stem cell. It is also known as the hemocytoblast, and it is derived from the mesenchyme. Then what happens is, this one pluripotent cell differentiates into 2 progenitor lines, the myeloid and the lymphoid. Now, proliferation and differentiation helps in the final formation of the blood cells. In addition to blood cells, the pluripotent stem cell also forms reticular cells, mast cells, osteocytes, and adipocytes. But as we have seen, the stem cell does not directly give rise to the final mature cell. Instead, it forms progenitor cells followed by blast cells. So now in the first stage, the pluripotent stem cell has given rise to the progenitor cells. This can be either myeloid or lymphoid. These progenitor cells are also called multipotent cells or colony forming units because they form colonies or clusters of cells. Now these colony forming units, depending upon the colony that they are forming, the group of cells that they are forming, can be CFU-E, colony forming unit E, which forms erythroblasts, colony forming unit GM, which forms a majority of the WBCs, that is the granulocytes and monocyte, and CFU-MAG, which forms the megakaryoblast or precursors to platelets. The myeloid progenitor line will give rise to majority of the WBCs and the RBCs, and it gets formed in the red bone marrow. The lymphoid progenitor line gives rise to lymphocytes, but it gets formed in the bone marrow just like myeloid. However, it matures in the lymphatic tissue. Disorders of the myeloid line give rise to myeloproliferative disorders, whereas disorders of the lymphoid line give rise to lymphoproliferative disorders. For example, acute lymphoid leukemia is because of the second category, and because of the first category, that is myeloproliferative disorders, we encounter diseases such as acute myeloid leukemia and the Philadelphia chromosome, which is also seen in the same category. Next, the progenitor cells are rendered incapable by giving rise to precursor cells. So, precursor cells are cells whose destiny is decided. That means a promonocyte has to form a monocyte. However, one step before, which was the progenitor cell, could have any fate. But once that progenitor cell is becoming a precursor cell, its destiny becomes fated, because all the precursors lead to only one direction. Promonocyte will lead to monocyte, band neutrophil will lead to neutrophil, etc. Once these become blast cells, they have microscopic characteristic qualities. So, once it becomes a band neutrophil, that band neutrophil will have the qualities to become the neutrophil finally. These blast cells are formed by proliferation of the progenitor line. The blast cells on the lymphoid side include the lymphoblasts, which will give rise to T cell, B cell and natural killer cell, and on the myeloid side they include blasts for the WBCs, which are called bands: band neutrophil, band eosinophil, band basophil, promonocyte and mast cell precursor. When it comes to the platelets, the precursor will be the megakaryoblast, and when it comes to RBCs, the precursor will be the erythroblast. Several chemicals and hormones regulate the process of differentiation and proliferation of blasts, the formation of blasts from the progenitor cells, and these are called hematopoietic growth factors. Of course, then the final step is the formation of the blood cells, which includes RBCs, WBCs, platelets and lymphocytes, etc. Now, what happens is the lymphoblasts will give rise to the natural killer cell, the B cell and the T cell, which are all now mature. In the last step, they are completely mature. Then on the myeloid side, you will have your RBCs, your monocytes and your mast cells, all your WBCs, neutrophils, eosinophils and basophils, all your granular WBCs, and you will also have your RBCs. Now, the regulation of haemopoiesis is under certain chemicals and hormones, as we discussed, which are called haemopoietic growth factors. Number one is erythropoietin. Erythropoietin increases the number of RBC precursors, erythrocyte precursors, because that's its function. It encourages the stem cell to differentiate more into the red blood cell line. Now, these red blood cell precursors produce more RBCs. In case of kidney failure or disorders of the kidney, erythropoietin is decreased, or the production of erythropoietin goes for a toss. This is because erythropoietin is produced by the kidneys. In such conditions, what happens is the number of RBCs decreases. As a result, the amount of oxygen that is available to the tissues to be transported to the tissues also decreases, and the function of the entire RBC, that is transport of oxygen, is hampered. Another hormone is the thrombopoietin. Thrombopoietin regulates the formation of platelets from the megakaryoblast, and it encourages the stem cell line to produce more platelets. The third would be cytokines. Cytokines are small glycoproteins that are produced by various cells, fibroblasts, endothelial cells, macrophages, WBCs, etc. Now, these are of two types. Cytokines are either interleukins or your colony stimulating factor. Now, these help in proliferation of the progenitor cells to form more WBCs. Hence, they account for two important functions, that is phagocytosis and also for accounting for cell-mediated immunity of the human body. Now, because these are responsible for formation of WBCs, in infections, interleukins go up. Interleukin is a kind of cytokine. Now, can you tell me a recent infection in which a very potent marker is interleukin 6? Yes, the coronavirus infection is often represented in the form of a high interleukin count.